We know that the amino acids are deaminated, producing ammonia. Ammonia is the toxic part. It is replicated in a process which is called urea cycle. Ammonia is toxic, urea is non-toxic. So the urea cycle is the main pathway of ammonia detoxification. But there are some additional pathways of ammonia detoxification. Some amounts of ammonia interact with glutamate, producing glutamate. This is glutamate. It interacts with ammonia with participation of ATP as energy source. The ammonia is bound to carboxylic group forming glutamate. So the carboxylic group is converted into amide group. The reaction is catalyzed by glutamine synthetase, not synthase, but synthetase, glutamine synthetase. So under the action of glutamine synthetase, ammonia interacts with glutamate, producing glutamine. Glutamine is a protein amino acid. It's a very important substance. Ammonia participates in regulation of pH of tissues and physiological fluids. For instance, in kidney, the glutamine is submitted to the action of glutaminates. With participation of water molecule. The water molecule hydrolyzes the amide bond. The amino group is released in the form of ammonia. Glutamine is converted back into glutamate. So please remember that glutamine synthetase converts glutamate into glutamine, and glutamate is an enzyme which converts glutamine into glutamate. The ammonia is excreted with urine. It neutralizes an excess of acids in human body and in body fluids. The other pathway of glutamine metabolism is biosynthesis of asparagine. So glutamine may interact with aspartate. Under the action of asparagine synthetase, with participation of ATP, which is converted into AMP and pyrophosphate, the amino group from glutamine is transferred to aspartate, producing asparagine. This is the pathway which synthesizes asparagine. Asparagine is degraded by the action of asparaginase. Asparaginase is an enzyme which hydrolyzes the amide bond in asparagine, converting it into are made with a release of ammonia. And now let's talk about the metabolism of hydrocarbon skeletons of amino acids. The metabolism of hydrocarbon skeletons of amino acids. So, some amino acids are converted into glucose. They are called glucogenic amino acids. Some of them are converted into ketone bodies. They are called ketogenic amino acids. Some of them are converted both into glucose and to ketone bodies. They are called glucoketogenic amino acids. At the beginning, let's look at the metabolism of glucogenic amino acids. The typical glucogenic amino acid is aspartate. Aspartate is submitted to transamination. The amino group is removed and transferred to a ketone acid, and the aspartate is converted into oxalacetate. You already know that the oxalacetate is a metabolite of gluconeogenesis. So oxalacetate enters the glucogenesis process, converted into glucose. The next typical glucogenic amino acid is alanine. So alanine is transaminated. It converts into pyruvate. Pyruvate is carboxylated, converted into oxalacetate, and oxalacetate is a metabolite of gluconeogenesis. It converts into glucose. This is the next typical glucogenic amino acid. The next glucogenic amino acid, typical glucogenic amino acid, is glutamate. Glutamate is submitted to transamination or to oxidative deamination. It loses amino group. It converts into alpha-ketoglutarate. Alpha-ketoglutarate. The alpha-ketoglutarate enters the Krebs cycle or citric acid cycle. It means the same. It is submitted to oxidative decarboxylation. This carboxylic group is removed in a form of carbon dioxide, and the alpha-ketoglutarate is converted into oxalacetate. Oxalacetate is a metabolite of gluconeogenesis. It converts into glucose. The next glucogenic amino acid is histidine. This is histidine. So the histidine, sorry, not histidine, but proline. Sorry. Proline is submitted to the action of proline dehydrogenase. Proline dehydrogenase is oxidized with participation of NAD+, which converts into NADH plus H+. The proline is converted into dehydroproline. This is the hydroproline. Then in spontaneous reaction with participation of water molecules, the dehydroproline converts into glutamate gamma semialdehyde. This is glutamate gamma semialdehyde. It has aldehyde group and carboxylic group. Therefore, it's called semialdehyde. Then, the glutamate gamma semialdehyde is submitted into glutamate semialdehyde dehydrogenase with participation of NAD+, which converts into NADH plus H+. Then, glutamate gamma semialdehyde is converted into glutamate. And you already know that glutamate is transaminated, it converts to alpha-ketoglutarate. Alpha-ketoglutarate is submitted to oxidative decarboxylation, and the product of this reaction is oxalacetate, which is a metabolite of gluconeogenesis. These are the typical glucogenic amino acids. The next group of amino acids are ketogenic amino acids. There are two ketogenic amino acids. There are two ketogenic amino acids, leucine and lysine. Let's look at the beginning and the metabolism of leucine. So leucine, this is leucine, carboxylic group amino group. It is submitted to transamination. The amino group is transferred to an acceptor. The leucine is converted into alpha-keto isocaproate. Alpha-keto isocaproate. Alpha-keto isocaproate. Then it is submitted to oxidative decarboxylation with participation of coenzyme A and NAD+. NAD+ converts into NADH plus H+. The carboxylic group is released in the form of carbon dioxide, and the coenzyme A interacts with the rest of molecule, forming isovaleril coenzyme A. Isovaleril coenzyme A. Isovaleril coenzyme A. This is energy-rich bond. Then the isovaleril coenzyme A is submitted to the action of isovaleril coenzyme A dehydrogenase, which cooperates with fat. Fat reduces forming fat H2. This hydrogen and one of these hydrogens interact with fat, converting into fat H2. Double bond forms at this place, like you see it here. Isovaleril coenzyme A is converted into beta-methyl crotonyl coenzyme A. Beta-methyl crotonyl coenzyme A. What happens next? In the next step, the product of former reaction beta-methyl crotonyl coenzyme A is submitted to the action of beta-methyl crotonyl carboxylase. This is Polish name, sorry. Beta-methyl crotonyl coenzyme A carboxylase. Beta-methyl crotonyl coenzyme A carboxylase. ATP converts into ADP and inorganic phosphate. Carbon dioxide interacts with this carbon, forming carboxylic group. The beta-methyl crotonyl coenzyme A converts into beta-methyl galactonyl, beta-methyl, glutaconyl, coenzyme A. Beta-methyl, glutaconyl, coenzyme A. This metabolite is submitted to the action of beta-methyl, glutaconyl, coenzyme A, hydrolase. Beta-methyl, glutaconyl, coenzyme A, hydrolase. Beta-methyl, glutaconyl, coenzyme A, hydrolase. Water molecule interacts with the double bond, forming this carboxyl group, hydroxyl group, and this CH2 group. The beta-methyl glutaconyl coenzyme A is converted into beta-hydroxy, beta-methyl glutaryl coenzyme A. Beta-hydroxy, beta-methyl glutaryl coenzyme A. Then, this metabolite is submitted to the action of beta-methyl glutaryl coenzyme, beta-hydroxy, beta-methyl glutaryl coenzyme A lyase. Beta-hydroxy, beta-methyl glutaryl coenzyme A lyase. Under the action of this enzyme, the molecule of this metabolite is cleaved at this place, releasing acetoacetate and acetyl coenzyme A. Acetoacetate is a ketone body. Acetoacetate is a ketone body. It may be converted into other ketone bodies, like beta-hydroxy, butyrate, or acetone. So after the reduction of the ketone group results in formation for beta-hydroxy butyrate, the carboxylation of acetoacetate results in formation of acetone. This is typical ketogenic amino acid. The other ketogenic amino acid is lysine. This is lysine. Lysine has 12 amino groups, 1, 2, alpha amino group, and epsilon amino group. Lysine is also converted to ketone bodies, but its metabolism is very complicated, so we don't require the knowledge of this process. But please remember that lysine is converted to acetoacetyl coenzyme A. Acetoacetyl coenzyme A, which is an active form of acetoacetate. So please remember that leucine is converted into acetoacetate, whereas lysine is converted into active form of acetoacetate to acetoacetyl coenzyme A. The third group of amino acids are glucoketogenic amino acids. Glucoketogenic amino acids. What does it mean? It means that they are converted into both glucose and ketone bodies. There are two glucoketogenic amino acids. Phenylalanine and tyrosine. Please look at this process. So in the first step, the phenylalanine is submitted to the action of phenylalanine hydroxylase. Phenylalanine hydroxylase. It cooperates with tetrahydrobiopterin. Tetrahydrobiopterin is converted into dihydrobiopterin. Oxygen molecule participates in this process. Water is formed. The oxygen incorporates between this carbon and the hydrogen, forming this OH group. So phenylalanine converts into tyrosine. What happens next? In the next step, the tyrosine is submitted to the action of tyrosine transaminase or tyrosine aminotransferase. Tyrosine transaminase or tyrosine aminotransferase. Alpha-ketoglutarate, sorry, this is Polish name. Alpha-ketoglutarate is converted into glutamate. Alpha-ketoglutarate is converted into glutamate. The amino group is removed. The tyrosine converts into para-hydroxyphenylpyruvate. Para-hydroxyphenylpyruvate. What happens next? In the next step, the product of former reaction, para-hydroxypyruvate, is submitted to the action of para-hydroxyphenylpyruvate hydroxylase. Para-hydroxyphenylpyruvate hydroxylase. This carboxylic group is removed in a form of carbon dioxide. The hydroxyphenylpyruvate is converted into homogenizate. One hydroxyl group, another hydroxyl. Then the homogenizate is submitted to the action of homogenizate oxidase. Homogenizate oxidase. But it's better to say homogenizate deoxygenase. Homogenizase deoxygenase. Oxygen molecule participates in this process. This double bond is cleaved two oxygen molecules are incorporated. 1, 2. The homogenizate is converted into malonyl acetylacetate. Malonyl acetylacetate. What happens in the next step? In the next step, the product of former reaction malonyl acetylacetate under the action of malonyl acetate isomerase is converted into fumarate acetylacetate. This is fumarate group, the red one, and this is acetylacetate group, the black one. This is the name of enzyme. So under the action of this enzyme, the malonyl acetylacetate is converted into fumarate acetylacetate. Then this metabolite is submitted to the action of fumarate acetylacetate hydrolase. Water molecule participates in this process. This bond is clipped, forming carboxylic group here and methyl group here. So the OH group from water binds to this carbon, forming this carboxylic group. This carboxylic group. Whereas the hydrogen from water binds to this carbon, forming methyl group. So the fumarate acetylacetate, under the action of fumarate acetylacetate hydrolase, is converted into fumarate acetylacetate. Fumarate is a metabolite of Krebs cycle. So it enters the Krebs cycle. It is converted into malate. Then malate is oxidized, forming biologically active substances produced from amino acids. The first active substance produced from amino acids are one carbon, active one carbon fragments, active one carbon fragments. Active one carbon fragments are active methyl group, active formyl group, active methyl group, or active formyl group. How the active formyl groups and active methyl groups are formed in human body? So, the source of one carbon fragment is serine. This is serine. CH2OH, CHNH3+, and carboxylic group. Please look at this red marked fragment.
Oh, shit.
So the red fragment is transferred from serine onto tetrahydrofolate. So this group is transferred to nitrogen 10 of tetrahydrofolate. It binds to this nitrogen, forming hydroxymethyl. You may call it hydroxymethyl tetrahydrofolate, or simply hydroxymethyl tetrahydrofolate, hydroxymethyl tetrahydrofolate, or hydroxymethyl tetrahydrofolate. What happens next? In the next step, the product of this reaction is decylated this OH group interacts with this hydrogen, forming water molecule. CH2 group appears. The CH2 forms a bridge between nitrogen 5 and nitrogen 10. Then the N5 and 10 methylene tetrahydrofolate may be submitted to the reaction of N5 and 10 methyl tetrahydrofolate reductase or N5 and 10 methyl tetrahydrofolate dehydrogenase. Reductase, dehydrogenase. N5 and 10 methyl tetrahydrofolate reductase and 5 and 10 methyl tetrahydrofolate dehydrogenase. In the first reaction, the CH2 group is converted into SH3 group, methyl group, bound with nitrogen 5. This is N5 methyl tetrahydrofolate. In this case, the SH2 group is oxidized, forming a group. So the reduction of these bonds results in formation of methyl group. The oxidation of this group results in formation of formyl group. This is catalyzed by reductase. This is catalyzed by dehydrogenase. Both N-methyl tetrahydrofolate and N5 formyl tetrahydrofolate are active one-carbon fragments. This is active methyl group. This is active formyl group. Please look again at the center. So this reaction is catalyzed by hydroxymethyl transferase, or hydroxymethyl methylene transferase, hydroxymethylene transferase, or hydroxymethyl transferase. This reaction is catalyzed by hydroxymethylene tetrahydrofolate dehydratase. This is catalyzed by reductase. This is catalyzed by dehydrogenase. Then N5 methyl tetrahydrofolate may serve as a donor of methyl group for methylene biosynthesis. So, the methyl group may interact with vitamin B12 with cobalamin. Here is omitted cobalamin and so cobalamin interacts with methyl group, converting into methylcobalamin. Methylcobalamin may interact with homocysteine, and the amino group, the methyl group, is translocated from cobalamin onto SH group of homocysteine, forming methionine. So, the material the N5 methyl tetrahydrofolate serves as a substrate in methylation processes. The formyl group serves in biosynthesis processes, in biosynthesis of purine, ring, in biosynthesis of thymine. The next group, the next biologically active substances produced from amino acids are catecholamines. Norepinephrine and epinephrine. So tyrosine is submitted to the reaction of tyrosine hydroxylase. Tetrahydrobiopterin is a coenzyme. Oxygen molecule participates in this process. One oxygen incorporates at this point, forming hydroxy group. The other oxygen atom interacts with hydrogen, forming water molecule. Tetrahydrobiopterin is converted into dihydrobiopterin. The reaction is catalyzed by this enzyme, tyrosine hydroxylase. Tyrosine is converted into dihydroxyphenylalanine or simply to deoxyphenylalanine. The abbreviation is DOPA. The OH groups appears at this point. DOPA may be converted, may be submitted to the action of DOPA decarboxylase. The carboxyl group is released in the form of carbon dioxide. The DOPA is converted into dopamine. What happens next? In the next step, the dopamine is submitted to the action of dopamine beta oxidase. Oxygen incorporates between this carbon and this hydrogen, forming this hydroxyl group. Dopamine is converted into norepinephrine. Then norepinephrine is submitted to the action of N-methyltransferase. So under the action of this enzyme, the norepinephrine is methylated. The donor of methyl group is S-adenosine methylene. I will tell you shortly about this coenzyme, S-adenosine methylene. S-adenosine methylene is a donor of methyl group. It replaces one of these hydrogens. Like you see it here. So the red fragment is transferred from S-adenosine methylene onto amino group or norepinephrine, converting into epinephrine. A part of DOPA is metabolized, are consumed in melanin formation. Melanin is a biological pigment which accumulates in skin, in eyes, in hairs, in central nervous system. It protects the body against the action of UV light, ultraviolet light. The next biologically active substances produced from amino acids are thyroid hormones. They are produced from phenylalanine and tyrosine. There are two hormones, 3-iodothyronine, T3, and tetraiodothyronine, or thyroxine, T4. This is T3, this is T4. 3-iodothyronine, tetraiodothyronine. So please look here, 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 and here. Here is iodine atom. So 3-iodothyronine contains 3 iodine atoms, whereas tetraiodothyronine contains 4 iodine atoms. The hormones of thyroid glands are the only biological substances which contain iodine. The next biologically active substance produced from amino acids, is nicotinic acid. As you already know, the nicotinic acid, it's better to say the amide of nicotinic acid, is a component of NAD plus and NADP plus. The nicotinic acid is produced from tryptophan. The metabolism is very complicated, so we will not require from you the knowledge of full metabolic pathway which produces nicotinic acid or nicotinic acid amide. Please remember that a part of tryptophan is converted into nicotinic acid amide or simply nicotinamide. The amide of nicotinic acid. The next biologically active substance which is produced from amino acid is cysteine. Cysteine, here E is omitted. Cysteine. This is cysteine. Carboxylic group SH. NH2 group, SH2, SH group. So, the cysteine is converted into cysteine sulfide. The SH group is oxidized, forming SO2 group. Cysteine is converted into cysteine sulfide. What happens next? In the next step, the cysteine sulfide is decarboxylated. The carboxylic group is released in the form of carbon is converted into hypotonic. In the next step, the SO2 group of hypotonic is oxidized to SO3 group, forming taurine. What is the biological function of taurine? Taurine interacts with bile acids, increasing its solubility, increasing their solubility. Furthermore, taurine accumulates in central nervous system. It works as neurotransmitter in central nervous system. The next biologically active substance produced from amino acid is active solvent. Active solvent. How the active solvent is produced? So, as you already know, the cysteine is oxidized, forming cysteine sulfate. So, not sulfate, sulfite, sorry, not sulfate, but sulfite. Cysteine sulfite is submitted to translation. The amino group is removed. Ketone group appears in this place. CO group appears. The cysteine sulfite is converted into beta sulfinyl pyruvate. Beta sulfinyl pyruvate. Very unstable substance, which is degraded by water molecule. So water molecule degrades this bond. This fragment is released in the form of pyruvate, and the red marked fragment binds oxygen, forming HSO3 minus product. Then the HSO3 minus ion is oxidized by oxygen with participation of water molecule. It is the SO3 group is oxidized to SO4 group. Water, one oxygen is incorporated into sulfate, the other incorporates to water, forming hydrogen peroxide. Proton is released. This hydrogen is released in the form of proton. What happens next? In the next step, the sulfate SO4, 2 minus, interacts with ATP. SO4, 2 minus, interacts with ATP, and under the action of adenyl sulfate transferase, ATP is split with the release of inorganic pyrophosphate, and the adenyl group interacts with sulfate, forming adenyl sulfate. Adenine, ribose, phosphate, sulfate. Please look here. This bond is highly energetic bond. What happens next? In the next step, the adenyl sulfate is submitted to the action of adenyl sulfate kinase. Under the action of this enzyme, ATP becomes a donor of phosphate. ATP converts into ADP, whereas the phosphate group is transferred to this OH group for the O phosphate group. The adenyl phosphate is converted into 3' phosphoadenosine 5 prime phosphosulfate, 3 prime phosphoadenosine, 5 prime phosphosulfate. This is so-called active solvent. It serves as a donor of sulfate group in detoxification processes and in biosynthesis of sulfated glycosaminoglycans, like chondroitin sulfate, like dermatan sulfate, like heparin, like heparan sulfate, dermatan sulfate. The next biologically active substance produced from your acid is S-adenosylmethionine. S-adenosylmethionine. How it's synthesized? So, the methionine interacts with ATP. ATP loses pyrophosphate and phosphate, and the adenosine interacts with this sulfate, forming such a product. Such a product is called S-adenosine methionine. Please look at this point. This sulfur is a donor of positive electric charge. It makes this methyl group very reactive. It may be easily transferred to various acceptors in a process which is called transmethylation process. So in transmethylation process, the SH3 group is transferred from S-adenosylmethionine to various acceptors. The S-adenosylmethionine is converted into S-adenosyl homocysteine. So please look at this sulfur. This sulfur does not bind to the methyl group. Then the S-adenosyl homocysteine is submitted to the action of water. The hydrogen from water binds to this sulfur, forming this SH group, and the OH from water binds to this carbon, forming this hydroxyl group. So under the action of water, S-adenosyl homocysteine converts into 3 adenosine and 3 homocysteine. Some amino acids participate in biosynthesis of other amino acids in a process which is called transsulfurylation. How this process looks like? So, transsulfurylation may produce cysteine from homocysteine and serine. So this is homocysteine. It contains SH group and serine contains OH group, hydroxyl group. So the SH group of homocysteine interacts with hydroxyl group of serine, the red marked fragment. They interact, the OH group interacts with hydrogen, forming water molecule. This sulfur binds to this carbon, forming cystathionine. Cystathionine contains a sulfur bridge between this carbon and this carbon. What happens next? Next, the cystathionine is submitted to the action of water, and the enzyme which is called cystathioninase. Water molecule participates in this process. It loses the action of these amino groups in a form of ammonia. This bond is split. This fragment is converted into alpha-ketobutyrate, and this fragment is converted into cysteine. Ammonia is released. The ammonia is a product of deamination, which proceeds at this place. So under the action of cystathioninase and water, the cystathionine is split, producing alpha-ketobutyrate and cysteine. This is trans-sulfur relation. It means that the sulfur is transferred. Sulfur is transferred from homocysteine onto serine, producing cysteine. What is the metabolic fate of homocysteine? So homocysteine, under the action of water, is desulfurated. It loses SH group, which releases in a form of SH2, hydrosulfur. The name of enzyme is wrong. This is homocysteinase. So under the action of homocysteinase, the homocysteine is converted into alpha-ketobutyrate, hydrosulfur is released, and the amino group is released in a form of ammonia. What happens with hydrosulfur? So the hydrosulfur is oxidized by oxygen. It is converted into sulfate. Hydrogen atoms are released in the form of protons. What happens with sulfur? Sulfur interacts with ATP, forming active sulfate. You already know this process. biologically active product produced in amino acid metabolism is gamma aminobutyrate. Gamma aminobutyrate. Gamma aminobutyrate is a neurotransmitter in central nervous system. It is produced from glutamate. So in the first step, the glutamate is submitted to decarboxylation. The carboxylic group of glutamate is released in the form of carbon dioxide. Glutamate is converted into gamma aminobutyrate. Gamma aminobutyrate. The relation is GABA. How the GABA is metabolized? It is submitted to transamination. It loses the aminobutyrate form of ammonia. The SH2 amino, SH2NH3, SH2 group is converted into aldehyde group. Gamma aminobutyrate is converted into succinate semialdehyde. Succinate semialdehyde. Then the succinate semialdehyde is oxidized. The aldehyde group is oxidized to carboxylic. Succinate semialdehyde is converted into succinate. Then the succinate enters the Krebs cycle, converted into fumarate. Fumarate is converted into malate. Malate is oxidized, forming oxalacetate, and oxalacetate is a metabolite of gluconeogenesis. It converts into glucose. The next biologically active substance is histamine. Histamine is produced from histidine. So the next biologically active substance produced from amino acid is histamine. So histidine is submitted to decarboxylation. The carboxylic group is released in the form of carbon dioxide. Histidine is converted into histamine. Histamine is a substance which dilates small arterial vessels. It decreases the arterial blood pressure. It increases hydrochloric acid production in gastric mucosa. It increases the permeability of endothelius. The next biologically active substance is putrescine. Putrescine is a product of ornithine decarboxylation. This is amino acid ornithine. This is its carboxylic group. So the carboxylic group is released in the form of carbon dioxide, and ornithine is converted into putrescine. So putrescine is a growth factor. Putrescine is a factor which neutralizes the negative electric charge of nucleic acids. Putrescine is a carrier of positive electric charge, therefore it easily interacts with nucleic acids, neutralizing its negative their negative electric charges. The next biologically active substance produced from amino acid is serotonin, or 5-hydroxytryptamine. It is produced from tryptophan. So tryptophan is hydroxylated with participation of oxygen, and tetrahydrobiopterin. So under the action of these compounds and the enzyme tryptophan 5-hydroxylase, the tryptophan is hydroxylated. Tryptophan is converted into 5-hydroxytryptophan. In the next step, the decarboxylated group is released in the form of carbon dioxide. 5-hydroxytryptophan is converted into 5-hydroxytryptamine or serotonin. 5-hydroxytryptamine is a substance which contracts the wall of small arteries. It increases arterial blood pressure. It aggregates blood platelets. The next biologically active substances produced from amino acids are intestine gas compounds, scatol and indole. They stimulate the peristaltics of intestine. They are produced from tryptophan. So tryptophan is dominated by bacteria. Xylem group disappears. Tryptophan is converted into indolopropionate. In the next step, the indolopropionate is decarboxylated. This fragment releases in the form of carbon dioxide, and this carbon is oxidized, forming carboxylic group. Indolopropionate is converted into indoloacetate. Indole group, acetate group. Then it is decarboxylated, converting into scatol, and scatol is demethylated, converting into indole. Both indole and scatols are gases which stimulate peristaltics of intestine. The next biologically active substances produced from amino acids are spermine and spermidine. They are polyamides. So, adenosine ATP interacts with methionine, forming S-adenosine methionine. S-adenosine methionine is decarboxylated, forming S-adenosine thiopropylamine. S-adenosine thiopropylamine. S-adenosine methyl thiopropylamine, which interacts with putrescine. Putrescine, under the action of spermidine synthase, converts into spermidine. So this group is transferred from S-adenosine methyl thiopropylamine onto putrescine forming spermidine. The other fragment releases in the form of methylthioadenosine. Then spermidine interacts with next molecule of S-adenosine, methylthiopropilamine, under the action of spermidine synthase, converted into spermidine. Methylthioadenosine is released. So both spermidine and spermidine are polyamides. They are carriers of positive negative charge. They neutralize the negative electric charge of nucleic acids. This is spermidine. This is spermidine. This is spermidine. Both of them play similar biological function. Thank you.
Thanks.